I've said this before, but viruses really don't fit into the definition of what it takes to be a living organism. So we know that they can't carry out metabolism. They don't reproduce on their own. They don't have a development process. So they're not fitting into any of that criteria. So they really don't consider viruses to be alive. They're more inert, where they're just sort of in this inactive stage for a period of time and they're always seeking a host. Since viruses can replicate only within cells, they probably evolved as bits of cellular nucleic acid. And there's volumes of books on where did these things come from, where did they begin, and part of one of their theories is that they actually are pieces of our own cells that turned against us because when they really look at them, sometimes the nucleic acid of the virus matches the nucleic acid of the host. So if that's the case, then it only stands to reason that they had to have been part of that host at some point. The candidates for viral genomes, now they're talking about plasmids and transposons. We haven't talked about transposons. Plasmids, going back to bacteria, is that circular piece of DNA in the bacteria that actually gets transferred into a new bacteria during the process of conjugation. Remember, the bacteria line up, they form a conjugation tube between them, and the plasmid, which is a separate little circular piece of DNA, gets transferred through that conjugation tube to the new bacterium, and now we have genetic variability. So this is what a plasmid is, and it's very mobile. It means it can move. Well, transposons are actually what are called jumping genes. And for whatever reason, we didn't really get a chance to talk about these when we were doing genetics. But there are genes that, for whatever reason, are capable of jumping from one location on the gene to another location. And when one gene jumps onto another, it deactivates whatever it lands on. I mean, just think about maybe a great big fat person come flying out of the air and landing on you. Well, it's going to shut you down, and whatever gene lands, is going to take over and is going to be expressed. Barbara McClintock, and I'm not going to ask you this, but just FYI, Barbara McClintock was the woman that discovered genes that jump. I am not certain of any genes that jump in humans, but I do know that one of the most famous of the examples of your jumping genes is in Indian corn, where you have got the different colors in Indian corn, and it's because some of the genes are considered to be transposons. But this last sentence says plasmids, transposons, and viruses, they're all genetic elements they're sort of entities on their own, but they're all very mobile. They're movable. And certainly depending on how you pick up a virus, it can be mobile. Now, they're not exactly moving on their own, but they're trying to gain entry. And don't forget that the glycoproteins on the coat of that virus have to match the proteins, the receptors on the outside of your cell, because the marker's first, the receptor, and then the channel. Not all viruses are going to enter through a channel. A lot of them are going to be binding to the receptor, and then your cell sort of engulfs them. Also, all of our viruses are what are called obligate intracellular parasites. And I believe this was on a PowerPoint that I may have skipped over. But obligate means they have to. They're directed to. They have to. Intracellular means they have to be taken within the cell, and of course they're parasitic. So we know that all animal viruses are obligate intracellular parasites. They're heterotrophic. They cannot reduce or metabolize on their own. They need all of our genetic machinery to make their babies, and they're all parasitic. There's not going to be a virus around that's going to be a good virus.